Classic. Okay, students, so welcome back. So before moving further, I'm showing you a video. So that video will further explain you the concept of ligation and how really you have to perform your experiment. So just wait for a moment.
It's 3 o'clock.
Trying to ligate a new gene into an old plasmid. Let's begin by discussing the reaction mechanism for ligation. First of all, any ligation reaction requires a ligase enzyme, ATP, and some DNA fragments. Ideally, the DNA fragments should have sticky ends. Sticky ends will greatly accelerate the rate of this reaction. It is possible to ligate fragments with blunt ends, but the reaction can take up to 10 times as long. Either way, the first step in ligation is for the ligase enzyme to form an intermediate complex with ATP by cleaving off two of the phosphates. The enzyme then transfers the AMP to a hydroxyl group on the phosphodiester backbone. This makes that hydroxyl group vulnerable to attack by the phosphate group on the other DNA fragment. Once that phosphate group attacks, the AMP falls off, and we have a sealed phosphodiester backbone on one of the DNA strands. The other strand of the double helix will have to be repaired in a subsequent reaction. So you can see here that ATP is a key component of this reaction. Without ATP, no ligation will occur. There are also two main steps in this process. You have an initial annealing step where the strands find each other and form hydrogen bonds. Then we have this enzymatic reaction with our ligase that permanently seals the phosphodiester bond. Both of these steps must occur in a successful ligation reaction. Unfortunately, though, they're favored at different temperatures. The initial annealing step is favored at low temperatures. That's where hydrogen bonds are the most stable. But at this low temperature, the ligase enzyme is very slow. Instead, the ligase enzyme prefers to work at 25 degrees C. Obviously, a higher temperature is going to favor enzyme activity. But at this higher temperature, your hydrogen bonds might be less stable. So we have this clash in optimum temperatures between the annealing step and the ligation step. And this gives us a few different options. You can choose to run the reaction at 4 degrees C, but give it a lot more time. Or you can run the reaction at 25 C for a shorter amount of time and anticipate having a slightly slower yield. However, most people just compromise between the two temperatures, find an average somewhere around 16 C, and let the reaction run for an hour. This gives you the best of both worlds and usually gets a pretty good yield. Alright, so now that you understand how ligation works, let's actually set up a ligation reaction. To do so, you need the following reagents. You're going to need the two fragments you want to ligate together, a ligation buffer containing ATP, and the T4 DNA ligase enzyme itself. Remember to keep the enzyme cold whenever possible. If you haven't already done so, you'll need to quantify the concentration of DNA in each sample. This is because ligation reactions work best when there's a 4:1 molar ratio of insert or gene DNA to backbone or plasmid DNA. So you'll either need to use a nanodrop or a plate reader to quantify the DNA concentrations in each sample. Make sure you use a blank, such as water, that matches the solvent in each of your DNA samples. Once you have the backbone and insert DNA concentrations, we can use these values to estimate how much volume of each sample we should add to our ligation reaction to ensure that we have that 4:1 molar ratio of insert to backbone DNA. You'll also need the number of base pairs in each fragment. You can find these values by looking up the sequences of your plasmid and your gene, or you can just roughly estimate them. You'll also need a molar ratio of insert to backbone DNA. We're probably going to use 4, but there's some other options we can discuss later. Once you define all these variables, though, you can calculate the volume of backbone and insert DNA to add to the ligation reaction using the equations shown on the right. But first, let's discuss a few guidelines for these variables. First of all, for your molar ratio of insert to backbone, we're going to use 4 today, but you can use anything between 4 to 10. Going any lower than this will significantly decrease your yield, but it might still work. Also, the concentration in each DNA sample should be approximately 2 to 10 nanograms per microliter. If your DNA concentrations are lower than 2 nanogram per microliter, then it's pretty unlikely that your ligation will work. If your concentrations are higher than 10 nanogram per microliter, then that might be simply too much work for the enzyme, and you'll end up with a lot of partially ligated plasmids, which is not ideal. But remember, if your DNA concentration is too high, it's pretty easy to dilute it down with water. Finally, if you look at the equations on the right, you'll notice we also need a total DNA volume. In the reaction we're performing today, we're going to use a total DNA volume that's backbone and insert mixed together of 16 microliters. This is pretty common. You don't need very much, but you can definitely go up or down from here as you wish. So once you've defined R and modified your concentrations as necessary, you can calculate VI and VB using these equations right here. To illustrate how this is done, let's walk through a quick example. Let's say you quantify your DNA concentrations and you have 6 nanogram per microliter of backbone and 8 nanogram per microliter of insert DNA. The plasmid backbone is 5250 base pairs long and the insert is 750 base pairs long. And we're going to stick with a 16 microliter total DNA volume. If we plug and chug all these values into our equation for insert volume, we get a value of 4.8 microliters. Notice here that I'm using a 4:1 molar ratio, so R is equal to 4. To obtain the volume of backbone DNA, I simply take 16 and subtract the insert volume. So here I get 11.2 microliters. So in this reaction, I'll be taking 4.8 microliters of insert DNA, 11.2 microliters of backbone DNA, and then I'll need to add my ligase buffer and ligase enzyme. In this reaction, I'm going to add 2 microliters of DNA ligase buffer, since it's supplied in a 10x concentration, and 2 microliters of T4 DNA ligase enzyme. And there you have it. Now we have the recipe for our reaction. Let's go ahead and set it up. Begin by mixing your insert and backbone DNA samples. For example, if you obtain the values shown in the previous example, you would be mixing 4.8 microliters of insert and 11.2 microliters of backbone DNA. Next, add 2 microliters of the T4 ligase enzyme buffer. And finally, add 2 microliters of the enzyme itself. Please, once you're finished with the enzyme and buffer, put them back in the freezer as soon as possible. Once your samples are well mixed, you'll need to incubate the reaction. If this is the last thing you're doing during the day, throw it in the fridge and let it sit overnight. If you're in more of a hurry, you can transfer your reaction to a PCR tube and incubate it at 16°C for 45 minutes in the thermal cycler. Finally, if you're feeling lucky, just leave the tube out at room temperature for about 30 minutes. Any one of these incubation strategies may work. However, if this is the first time you're doing this, you might want to split the sample up and try all the different incubation temperatures and see what works best for your reaction. Either way, once your incubation step is complete, you're ready to proceed to transformation, where we're going to use this ligated DNA to transform some E. coli bacteria. Hello! In this video, we're going to show you how to ligate 2 fragments of DNA together. You're trying to ligate a gene into a plasmid.
Okay, so hope you all have enjoyed this video. Am I audible to all? Am I audible to all? Am I audible to all?
Yes, sir. Yes, sir.
Okay, so
Yes, sir.
So this video, I think, able to explain you all the aspects. So what volume, how to calculate the volume, how to calculate in what ratio you need to mix them, and what concentration of your DNA you are going to use, and why we are keeping temperature low, like 35 degrees, 37 degrees Celsius. Mentioned is the optimum temperature, but as I mentioned, we are using 16 degrees Celsius for ligation. And reason behind is the efficiency is based upon how close these two sticky ends come together, and they should stay together. This sticking is because of the hydrogen bond, and hydrogen bond is weak bond, but it's become a bit stronger if temperature is going to be low. Because thing is that when you increasing the temperature, with the increase in temperature, you are increasing the kinetic energy. So kinetic energy means Brownian motion of your molecule within your solution. So molecules, they are not staying. When so you're saying solution doesn't mean your molecules are staying as static in the solution. Molecules are going to migrate. They are in a random motion throughout the solution. So, but you want them to come together, to stay together, so that you can do if you decrease the kinetic energy. So kinetic energy, if you lower down the temperature, so as you know that in case of gases also, if you keep on lower down the temperature, the volume of your gas keeps on coming down, and you can get it liquefied. Liquefy in the more less movement of your parts, and the solid still they become stable. So there's a thing we are doing here also. when we are decreasing the temperature, we are minimizing the kinetic energy, and the kinetic energy is going to be low. The strength of hydrogen bond is going to be high. But thing is that as we know that the optimum temperature is going to be 37 degrees Celsius, so we can't have a temperature 4 degrees Celsius, the enzyme is going to become inactive. So we need an in-between thing so that we can have some of the hydrogen bond and some of your ligation activity also. So for that we the beach ka rasta, so we choose a mid path, both the side. So if in the mid path, your incubation time is going to be increased. So that's why we are incubating overnight or around 16 hours of incubation to get your complete ligation. So in case of your blunt end, you further need even a longer time. So in the blunt end, you may argue that sir, blunt end there is going to be no hydrogen bond in them. Then can we go with the room temperature with the blunt ended one? Clear answer is also going to be no. Thing is that again the kinetic energy issue is going to be there. So generally for both we keep mid temperature type so that with less of the kinetic energy chances are there that two molecules that you want to ligate, they should come close to each other and they are going to be done. So wait for a moment. So I'm bringing you some video for ligating blunt end. So I didn't get a nice video which describe you for the ligation of your blunt end sequences. Generally, the videos that are available, they're showing the ligation using your linkers or adapters that we already talked about when we were talking in your theory. So, so in your experiment, if you want to perform your blunt end ligation also. So in that case, only the precaution that you need to take care about the temperature. So in case of the blunt end ligation, when you want to go for, so instead of 16 degrees Celsius, you further lower down the temperature. So you can go with the 4 degrees Celsius. So then you will say that the enzyme is going to be functional at the 4 degrees Celsius. So answer is yes, it will be functional, but it is not as much active as it was at 37 and at 16. Now the time you need further incubation time is going to be further higher. So if you are ligating at a 4 degrees Celsius, this simply means you have to incubate for longer. Simply you can say in 16 degree, a 12-hour incubation is going to be efficient for you. But now in 4 degree, you may need to go for the 16 or 18 or 24 hour of the incubation. And then how to confirm the ligation is going to happen, ligation is happening or not, that you have to do simply by turning on your electrophoresis and or moving for the transformation. Okay, so with the let me just check also your VLab. Is any something available at the VLab portal that we can also check? Is they are also having any? Okay, hope you are able to see the VLab portal also. So this is showing what this ligation is going to be. So you can get information from here also. So how you're going to perform. So what you need 3' hydroxyl and 5' phosphate. Gene of insert, foreign DNA. You have to like it acid digest to take out the gene of interest, mix with your vector and transform. And this all the image I already shown you. So how this is going to happen. And how this T14 ligase is going to work. This also is already there. You can read it here also. Application also if you want to know about. So ligation of cohesive or blunt end fragments for cloning, annealing, NICs, and double-stranded DNA. Ligation of synthetic linker and blunt ends that you can do. This calculation we already discussed about it. In the procedure, whilst that all these that you need, reaction mixture or distilled water, deionized water, DNA ligase, vector, insert, buffer, etc. All these things you need. So how to perform this experiment? Take a clean, fresh microfuse tube. Microfuse tube, then add all the requirement things. Most important thing is incubation. So here if you see the incubation at the room temperature, 22 degrees Celsius. So then you need a lesser time, that is 2 hours. But if you want to go for a lower temperature, then you need to go for more than 2 hours, a longer time duration. So you're analyzing it by the transformation. So what is really going to happen when you're performing your experiment in the lab? So the TNA for like contain ATP, that's the precaution that you need to check. So repeated freeze thaw you should not go for, that we already discussed in our method. So better vortex or spin, so that better the pipetting. so that everything settle down. So that's why it's better to vortex and followed by the screen. And animation, I don't think it will be working here. So animation, you can go to vmrta.edu, so you can get it there itself. And some assignments are also there if you want to go to perform something. So you can go with these assignments, these questions, you can try their answers. And for reading also, so these are the links, so you can get it, the information about the ligation, all these sites. And NCBI book is there in it, so you can open it. So I'm opening the first one, whether it is available. Okay, so this is a book which is showing you, so all the components are there, so you can read it from here also. And if you wish to download, you can download the PDF of it. PDF might be available, so you can check where the PDF is. And you can read it. So what recombinant DNA restriction on nucleus is there. So lots of things are there. Vector recombinant DNA there. Sequencing is there. So lots of information are there. So you can get all this information from here. And I am also shared. I already shared you this protocol manual and the V labs. You can get it most of information that you need. And if you want to evaluate yourself, some MCQs are here also, so you can check. You can self-evaluate and submit it. You will get the marks. So just go for self-evaluation at this site also. So what you need for the ligation purpose, etc. So with this thing, I think online laboratory wise, we have done. And anything that you feel that you should ask regarding this experiment, and hopefully by next week you will be in the campus, so then we'll perform all these experiments one by one. And just a brief introduction what we are going to do in our next lab. So next lab is the next part of this experiment. As we mentioned, you want to confirm whether the transformant or whether the ligation is done or not. That you are confirming by the transformation. Means transforming a foreign DNA you are going to transfer into the host cell. So when you are transferring a foreign DNA, it usually do not happen. So no cell would not, like if I try to give you a thing which you do not want, like no, none of you want to come to the campus. Now you're enjoying at home, but now institute is asking you to come to the campus. So we are forcing you to come to the campus. You are not willing. So we are making you competent. We are making you so that you should come to the campus. So that is the process that we have to do with our host cell also, to make our host cell to take up the foreign DNA. So the process by which we make our host cell to take a foreign DNA in it is called making competent cell. So how to make a bacterial cell competent? Like generally, most of the case, as our theoretical part and theory also, we referred most of the time E. coli, whether we are referring an enzyme or so on. So host is going to be E. coli. So why E. coli is going to be the host? Because it's a model organism, model microbes. Most of things are known about it. Genome is sequenced. Everything with a 5,000 kilobase pair genome that it have. You know everything about it. So every gene is recorded. So you know it's non-toxic also. Toxic strain, you know that these strains are toxic, but we are working with a non-toxic strain. So that's what is going to be best for us. So how we are going to, and E. coli doesn't like to take the vector that you want to insert in it. And we need a host which should not have its inherent plasmid, because if it is having inherent plasmid, then that may interfere with your plasmid that you want to insert. because then compatibility issues are going to be there, and also cell would like to stay with its own plasmid, not with the foreign plasmid. Or one more property that, as I mentioned, that cell do not like to take the plasmid. So there is always a chance that the host cell will lose the plasmid. Like you have transformed it, you facilitate the cell to take the foreign DNA, but when it is growing, so it's not its own thing. If it is forcefully you are giving something, doing something, then whenever you will get a chance, like whenever you're coming to the class, so if you don't want to stay in the class, but for sake of attendance, and even here also, I don't know how many of you are listening. So as you are having a chance that you can just log into the Steam portal and you can leave and you can enjoy your talking or enjoy your all other activity, just keeping your mic in hand-free mode. So whenever I call your name, you will say yes, sir. Otherwise, you are enjoying your stuff. So in similar way, whenever your cell got chance to enjoy, so cell will lose the plasmid. So for that to maintain the plasmid, maintain the foreign DNA into your cell, we have to keep our cell always under pressure. So what pressure we should do? We have to keep that selection pressure is there. Selection means you are selecting only those which are having the plasmid should grow. Those cells which are leaving the plasmid, we don't want them, so they should die. So if you allow them to grow, then their population will keep on rising, and then the cell that you need, the clone that we are referring, the clone means a recombinant clone which have your gene of insert with your recombinant plasmid, is going to keep on down. So that screening issue is going to be there. So for that, always we keep under selection pressure. So selection pressure is going to be for your as that antibiotic pressure we keep. So we always grow our recombinant under the antibiotic condition. So we are not growing them in antibiotic free condition, because as you give the antibiotic free condition, so cells which have tendency to lose these vector, they will grow up, and you will reach to a level and none of your cell are going to have your plasmid vector. So the most of the population is having lost your plasmid. So for that, all these things we have to keep in mind. So this is just for today. So I will create an assignment for your this experiment also. This is the ligation. So you have to submit this by the next Wednesday. So you have time. So for return material, you have the protocol manual and also the VLab is there for you. So from there you can choose to write to have the protocol and write all the requirement, all the principles. And also one thing that I showed you, the 2 videos, and 2 videos in the mechanism way. Also this mechanism of the ligation is there on VLab portal also. So from that also you can get those images. So how really the ligase is going to work. So 3-step 1, ligase come up with at the lysine, go and attach with the ATP. Then with this ATP that you call the adenylated enzyme, this will go and transfer it to your 5' end, and then it is going to ligate that. So all this 3-step process that you also have to mention in your record book and that you have to upload. And one more thing to keep in mind, that from Monday onward you are coming to the campus. So all of you must have your record book with you. So whatever you have uploaded, that's fine. Okay, so I may give you excuse from uploading because from next Thursday we are going to be in the campus. So everything, whatever, all the practical that we have done, so everything must be in written form. Like if they are in the segregated pages with you, so you have to combine all and compile in a single file. They must not be in loose form. It's not like just you come up with these loose pages to me and throw on my face that, sir, this is my practical record book. So don't do like that. Just have in a properly bound way. If you are having a file, must be tagged properly, and then hand over me in a gentle way. Don't throw on my face. Okay. So with that, I am concluding today's session. So please turn on your camera when I call your name. So one of you is absent. So who is absent? Okay, so I will now make the attendance list for that. Okay, so Adish.
Present, sir.
Atoiba.
Present, sir.
Ayushi.
Present, sir.
Demetrius.
Present, sir.
Devanshi. Devanshi. Ishita. Janvi.
Present, sir. Present, sir.
Kshitij.
Present, sir.
Manaswini.
Present, sir.
Namrita.
Present, sir.
Nandini.
Present, sir.
Nehal. Rupal.
Present, sir.
Sahiba.
Present, sir.
Sanjana.
Present, sir.
Shakti.
Present, sir.
Shivani. Tanvi.
Present, sir.
Shivani.
Present, sir.
Tridev.
Present, sir.
Vivek. Yukta.
Present, sir.
Yuvraj. So Nehal and Vivek.
Sorry, sir, I'm present. My mic wasn't working earlier.
कोई बात नहीं है, मैं समझ सकता हूँ ऑनलाइन में कैसा माइक काम नहीं करता. ओके, सो इट्स फाइन. सो विवेक भाग गया लगता है. ओके, सो थैंक यू ऑल. स्टे सेफ, स्टे हैप्पी. See you again on Friday. So our last online session. After that will be face to face. Okay, so on Monday our Monday session is going to be face to face. Okay, so stay safe, stay happy. Yes, Vivek also came back. Vivek also came back. अब ये कहाँ घूम के आए भाई?
नहीं, कहीं घूम के नहीं आया था सर. सॉरी, मेरा माइक कोई प्रॉब्लम चल रही थी जो पहले चल रही थी.
So Yuvraj and Yukta.
Yes, sir.
Okay, so see you on Friday. So stay safe, stay happy.
थैंक यू सर.
Sir, I'm present.
हाँ हो, सभी हो.